Okay, so measles virus, or paramyx, which is the monovirus used to study paramyxoviruses, multiplication cycle. So let's go through this. So in a textbook, Paramyxoviridae and Rhabdoviridae grouped together in one chapter because they share very similar multiplication cycles. So I expect this video to be much shorter than a Rhabdoviridae one. So let's just go through it. So here you have your Paramyxoviridae virion. Now there are two antigens on the Paramyxovirus. That is your hemagglutinin / neuraminidase antigen, and then you also have the fusion antigen. So the hemagglutinin neuraminidase antigens function in attachment and basically attachment to a cell. Sorry, just blinking out. And then the fusion, the F antigen, functions in fusing the virus to the cell. The way it works is F changes shape when your hemagglutinin, the fusion protein changes shape when the hemagglutinin neuraminidase antigen attaches. So F follows H, or fusion follows a hemagglutinin neuraminidase receptor, and see right here. And then you basically get attachment and fusion, right? Attach and penetrate, and then uncoat, and then you uncoat. And finally here's your uncoded genome, which is negative single-stranded RNA. So looking at the genome, just before we go further into multiplication cycle, genome native ssRNA, 15,000, basically 16,000 base pairs. It's pretty big. And you can see that the order is NPM-FHL. Now, Rhabdoviridae, I didn't say this in the video before, but the Rhabdoviridae gene order is NPM-GL, with G being a glycoproteins. So easy way to remember this is you see that this F and H, those are glycoproteins. So it's the same order as Rhabdoviridae, but now instead of the G is now turned to into an F and an H. The other thing to notice is that it doesn't just have a phosphoprotein. For Paramyxoviridae, or it doesn't have just one P protein there, like Rhabdoviridae. It has the P for Paramyxoviridae actually encodes for 3 different protein products using one open reading frame. And the way it does this is through multiple having 2 different start codons, with one start codon coding for P and the other coding for C. And to code for the V, to get the V protein, you have a frame shift during translation. So you have your native ssRNA genome here, right? And just very similar, pretty much exactly the same as Rhabdoviridae. You want to convert that native ssRNA to positive ssRNA. You transcribe the whole thing. And at the same time, you also have transcription of the individual reading frames. So you can see here, there's this thing, primary transcription. And just like Rhabdoviridae, Paramyxoviridae has virion-associated polymerases. So you can see that L and the P, those come with the virus. The L is a polymerase, the P is a thing that helps the L do its job. And then your nucleocapsid protein, like normal. And just like in Rhabdovirus, you have intergenic sequences and poly-U sites to help regulate what amounts of the genes you need more of. So you need a lot of structural proteins, your N protein. You need lots of your M protein. your matrix protein, but you don't need as much L polymerase. Right, and again, you get the idea. I have faith in you. So now you have this. Now you have a primary, like a basal level of translation, right? And now you make all these proteins, NPM, and NPM, and then your L and C. You also, of course, make your not your G proteins, but something analogous to it. Your H and F proteins. Again, compare measles and VSV, or compare Paramyxoviridae, Rhabdoviridae. They're exactly the same in the cycle. It's just slightly different protein names. So these pathogenic proteins all go up here. They go up here. They can do secondary transcription using the. They can use no. They can go up here, take that positive ssRNA that you made in the beginning, make a huge amount of your original genome. Now you have a bunch of your original genome. From here, you can have more of your transcription, and then you can have more proteins made. But then from here too, from here with this and all these amounts of native ssRNA, you can also replicate more. You can now make more positive single-stranded RNA intermediates to make even more native ssRNA, to transcribe more of this, to make more protein, At the end of the day, you have assembly. You have a bunch of protein, a bunch of viral proteins, a bunch of genome. You get particle assembly, and then done. Okay, so in studying Paramyxoviridae, there's a reason why it was grouped with Rhabdoviridae in the textbook, and it's also why we learned Paramyxoviridae right after Rhabdoviridae. They shared basically the exact same multiplication cycle, albeit with a slightly different protein names. One last note is for this PVC thing. In the lecture video and the video that I made, go back and look at that, or look in the textbook. You'll find out how V and C are made from just one open reading frame. Alright, so we're going to move on to Orthomyxoviridae next.